Alright guys, welcome back to Underground Science. In this video, we'll be covering complex 4 of the electron transport chain. Alright, so if you haven't watched the other 3 parts of the electron transport chain where I cover complex 1, 2, and 3, go and check those videos out. Alright, let's go and get started. Alright, so remember, just a refresher that the electron transport chain is occurring on the cristae, or the inner mitochondrial membrane. And so let's go ahead and just a really quick refresher so this video is not too long, is that I have here a short screenshot picture of what we did in the previous videos of electron transport chain. So here we see complex 1 or NADH dehydrogenase coenzyme Q oxyreductase, right? We'll just refer to as NADH dehydrogenase, sorry about that. And we saw that we have iron sulfur clusters here, and we also saw that NADH got oxidized into NAD+, right? And we also released some hydrogen protons in the vicinity, right, in the mitochondrial matrix, which is right here, all this space. And so what happened was that when we got oxidized here, we actually lost 2 electrons because of the oxidation reaction that NADH dehydrogenase did on the NADH. And so those 2 electrons then we said went to flavin mononucleotide, all right, or FMN. Flavin mononucleotide got reduced, turned into FMNH2, gained its 2 hydrogens, right? And then we said FMNH2 or flavin mononucleotide H2 then transferred those 2 electrons to the iron sulfur clusters, and then the iron sulfur clusters, couldn't speak there, transferred its 2 electrons finally to the coenzyme Q, right, or the ubiquinone. When we said that, a reminder, the coenzyme Q is not made up of protein, right? And when we're talking about the electron transport chain, and it's mobile. So that was complex 1. And we said that when we transfer those electrons, we said we lost energy, right? We released energy, not lost energy, we released energy. That release of energy was used to pump 4 hydrogen protons out into the intermembrane space via a pore that's present in this complex 1. Complex 2, we said, was a little different. We said complex 2 involved FADH2, another FADH2, right? Another electron carrier that we made, right, in the Krebs cycle. If you guys remember from when we went from succinate to fumarate, right? So we used succinate dehydrogenase for that. And remember we said succinate dehydrogenase was the only enzyme in the Krebs cycle that was bound to the cristae because it's also involved in the electron transport chain, right? It's also called complex 2. And we said that succinate actually got oxidized, lost 2 hydrogens, and each hydrogen atom has 1 electron, so lost 2 electrons. Those 2 electrons then, what happened was in the electron transport chain, we said the opposite happens. FADH2 gets oxidized into FAD, lets go of its 2 hydrogen protons, gets oxidized because it loses its 2 hydrogens, which has 2 electrons in total. Those 2 electrons then gets passed to the iron sulfur clusters present in the succinate dehydrogenase complex 2 protein, and then we said that coenzyme Q, right here, so picks up those 2 electrons from the iron sulfur clusters in the complex 2, and then that's it, right? But what did we say that was important right here is that when we do release energy, that energy is not used to pump protons out because complex 2 does not have any pores, and also in general, it just doesn't pump any protons out into the intermembrane space vicinity. Okay, and then we made a complex 3 video, which we don't have shown in this picture, but make sure to go and check that video out. But in just a quick recap, in the complex 3, right? So let's just go and do a quick recap so we don't waste too much time. Is that complex 3 actually had, so actually I drew it in the wrong space because I just noticed the cytochrome B here. Let's go and draw it around like that. So let's say this is complex 3, right? Complex 3 was named, if you guys remember, coenzyme Q, cytochrome C oxyreductase. And it had a cytochrome B component inside it. The cytochrome B component, actually, we said that had iron bound to it in its 3+ oxidation state. And we said that coenzyme Q actually transferred those electrons to the coenzyme Q cytochrome C oxyreductase, or complex 3. Right, so this is a quick recap. This is complex 3, right? And so these electrons, as it got transferred, we said those Fe3 plus iron, right, atoms in the 3+ oxidation state, receive those electrons. Oops, not from there. Sorry about that. Not from there. From the coenzyme Q, right? And as they received those electrons, they went to the they kept on going to the 2 plus oxidation state, right? So Fe2 plus, and then we'll see in this video, right, along with complex 4 and cytochrome C, that then this iron that just got reduced gives up its electrons, right, one by one to cytochrome C, and then this iron then turns back into the 3 plus. So it's kind of like a cycle that repeats itself, and remember there's a lot of iron that's bound to the cytochrome B. And we also said that as we did that, as we transfer those electrons one by one to cytochrome C, we said we pumped 4 4 hydrogen protons out. Okay, so from the mitochondrial matrix, right, from the mitochondrial matrix to the intermembrane space. All right, and now let's go and talk about what happens next. Okay, so now let's go and draw out our cytochrome C, okay, because cytochrome C is before our complex 4. So let's go and say that this actually, you know what, in particular, it's really not part of our cristae. It's kind of up right here, all right? Sorry, trying to having a little trouble speaking to it, I don't know why, all right? But so this is our cytochrome C, so cytochrome C, all right? And so what cytochrome C is going to do is, remember that we have our complex 3 right here. Okay, so let's go and draw it in a different color. We have our complex 3 right here. And remember, we just said that complex 3 was also known as cytochrome C, or coenzyme Q cytochrome C oxidoreductase, right? So this is our complex 3. And then we said that had a cytochrome B component to it with a bunch of iron that's bound to it, right? Fe3 plus. And we said that it received electrons, right, from the coenzyme Q that had those electrons initially from complex 1 and 2. So let's go and draw the coenzyme Q here, like chilling over here, right? So let's go and draw the coenzyme Q chilling over here. And we see that the coenzyme Q is given those electrons, right, at its own pace. And so coenzyme Q is mobile. Remember, it's not made of protein. It's giving those electrons to the coenzyme Q cytochrome C oxidoreductase complex, or complex 3. And it's causing the Fe3 plus to be reduced to Fe2 plus and back to Fe3 plus. But when it goes, so here's the thing, right? So we have Fe3 plus, right? Let's say this electron comes here and it reduces the Fe3 plus, turning the Fe3 plus into Fe2 plus, because we just gained a negative charge. So the oxidation state has to change. Just got more negative, right? So it went to Fe2 plus. Now, once it's at Fe2 plus, we said that it has to go back to Fe3 plus. But how does it do that? It lets go of this electron, right? So it takes electron, lets go of it, and passes it on to cytochrome C. So this electron goes to cytochrome C. Now cytochrome C, we mentioned, I forgot what video we mentioned it, but I'm pretty sure we did, is that it's always telling cytochrome, or it's always telling the complex 3 to chill out. All right, so it's like, chill out, I can only take and transfer 1 electron at a time. All right, so remember, cytochrome C can only take and transfer 1 electron at a time. So it's going to take 1 electron at a time from our coenzyme Q cytochrome C oxidoreductase complex, or complex 3. It's going to take those electrons, right? And then what it's going to do is it's going to pass it to the last complex, right? Or technically last complex, okay? Because we also have another huge, amazing enzyme that's counted as complex 5, but let's just go and say this is technically the last complex, right? And the electron transport chain, right? The ATP synthase is going to be part of oxidative phosphorylation. So anyways, this electron that cytochrome C receives 1 at a time is then, the cytochrome C will then transfer this electron onto complex 4. Now let's see what complex 4 names is, or what the name of complex 4 is. So complex 4, guys, is called cytochrome C oxidase. All right, cytochrome C oxidase. And why is it called cytochrome C oxidase? Well, it's called cytochrome C oxidase is because it's saying I'm oxidizing cytochrome C, right? Cytochrome C is right here. This complex 4, so let's go and write this down, that this is also called complex 4, right? So this is also called complex 4. So it's also called complex 4. Let's go and write it out as a whole. And so it's called cytochrome C oxidase because it's oxidizing cytochrome C, right? And it's also because we'll see later that oxygen is going to be our last electron acceptor. So we can see these proteins are just accepting electrons, passing the electrons, accepting electrons, passing the electrons, right? So cytochrome C is going to oxidize the crap out of cytochrome C here, or cytochrome C oxidase, sorry, is going to oxidize the crap out of cytochrome C because it's taking away electrons from it, right? Technically, because a cytochrome C is going to be, it's going to be transferring those electrons to cytochrome C oxidase. So anyways, then the cytochrome C oxidase will transfer electrons one at a time to our complex 4. Now we have another cytochrome component in our complex 4, so we made up of cytochrome A, and cytochrome A3. All right, so that's not a good color. So let me go and write, nope, that's not a, dang it, running out of good colors here. So cytochrome, oh, sweet. All right, cytochrome A and A3. All right, now cytochrome A and A3 are components in there with they have copper bound to them and iron bound to them. Since we've been consistent with using the iron example, let's go and say that we'll have iron bound here, right? And as we pass these electrons on to these cytochrome A and A3, these iron molecules, or these iron atoms with the 3+ oxidation state, will pick up those electrons, turning into Fe2+, and then these iron atoms are then going to take those electrons, and it's going to pass it on to oxygen, right? In particular, atomic oxygen. So it's going to pass it on to atomic oxygen. And then that's going to lead to these iron 2+ state, going back to iron 3+ state. When it passes those electrons to oxygen, so again, what do we see here? That oxygen is the end. So complex 4 is the last complex before we pass on to electrons. Oxygen is going to be the last electron acceptor of the electron transport chain. That's why we need to breathe oxygen, right? Or we can't really make ATP without oxygen being there, because we need to have oxygen to be our last electron acceptor. So oxygen is going to be our last and final electron acceptor in the electron transport chain, right? So oxygen is just chilling there. He's waiting. Complex 4 is like, all right, I got you. I'll throw an electron at you, or all the electrons that I'm receiving, and you're going to accept those electrons. Okay, so oxygen, so I'm going to write this down, scoot it over a little here, is that oxygen will then accept those electrons, and what's going to happen is that, remember, there's still a bunch of hydrogen protons in this matrix vicinity. So 2 protons are then going to come and be like, yo, I got you. I'll come and join with you, and what am I going to make? Well, what's the formula of water? H2O, right? This is atomic oxygen. So they're going to join, so oxygen is going to get reduced, join with 2 hydrogen protons in the vicinity of the matrix, and make water. All right? And so that's what happens at the last electron transport chain part, where we talk about complex 4. All right, so again, oxygen is our last electron acceptor, joins with 2 hydrogens and a hydrogen cations and makes water. Now, before we forget, complex 4, as it passes electrons down, releases energy, and it pumps 2 hydrogen protons out into the intermembrane space from the matrix of the mitochondria. So it's going to represent that. So now it's going to pump, when it gives up those electrons, it's going to take 2 hydrogen protons, not 4 like complex 1 and 3. It's going to take 2 and pump it out into the intermembrane space. All right, so as you guys saw along these videos of the electron transport chain, we saw that, we saw that we were pumping a lot of hydrogen protons from the matrix to the intermembrane space. So we're seeing that the concentration of H+ in the intermembrane space is increasing like crap, all right? And that's what's going to play a big role when we talk about oxidative phosphorylation, where we talk about the ATP synthase enzyme, or also called complex 5, all right? Because then what's going to be involved is that the ATP synthase is going to take all the high concentration of protons, pump it back in through the ATP synthase enzyme, and that's going to help make a bunch of ATP through oxidative phosphorylation. Couldn't speak there again. All right, so that's, let's see, hopefully I'm not forgetting anything, but that's pretty much it, right? That's the main idea of the electron transport chain, is that, oh, one other cool thing to know is that if you're wondering how are the electrons how do they know to keep on going from one protein complex to another in this specific direction? And remember, things are getting reduced, things are getting oxidized, right? That's in the electron transport chain. The reason why electrons move in this direction in the order of complex 1, 2, 3, 4, and then soon the ATP synthase enzyme is because as we go from complex 1 to the end, we increase in the positivity of our redox potentials. So in the beginning, what happens, right? So let me go and write it down here. So as we go from left to right in the electron transport chain, we increase in our value. We increase in the value of our redox potentials. Now, what does that mean? So just a quick overview is that first we're more negative when we're starting in complex 1. All right, so it actually starts to be more negative, and the specific values aren't really important right now. I'm just trying to get the overview down. Is that we start being really negative. So let me represent it by magnitude. All right, so we start being really negative here, and then as we increase in complex and in proteins, right, that are responsible in the electron transport chain, we start to get a little bit more positive, and then we start to increase in positivity. So we go from very negative to less negative to less negative to a little bit positive and more positive as we go from complex 1 to 5, right? 5 being ATP synthase. If you recall, electrons are negatively charged, so they want to be attracted to the positively charged place, right? So that's why we keep on accepting and donating electrons in this order, in this fashion, from complex 1 to the end, because it gets more positive here and electrons are attracted to positive charges, right? So electrons, if you recall, have a negative charge, and there's a lot of negativity here, right, in terms of redox potentials. And so electrons don't like that, and they repel against negative charges, so they're going to be attracted towards they're going to keep on going along the proteins of the cristae, electron transport chain, and they're going to keep on getting attracted to the complexes as the complexes, right, the protein complex on the cristae increase, they're going to be attracted to that because the complex proteins also increase redox potentials in terms of positivity, and so pretty much electrons are more attracted to a positive charge. So that's why electrons go from complex 1 and keep on going until they reach the end, all right, to oxygen. Right, so oxygen has the highest positivity for redox potentials. That's why it ends up as oxygen being the last electron acceptor. All right, so I'm sorry if this was like too messy or confusing. I hope it wasn't. Okay, I can't talk today, but I hope this video made sense to you guys. Make sure to tune into my future videos where I'll talk about more cool topics about biochem, physics, and chem. And don't forget to hit that notification bell, like, and subscribe. All right, and see you later, and stay safe.